All right, so on the gel, we can't look for the BRCA2 gene. So what we're going to have to do is find something else called a marker. All right, so markers are non-coding pieces of DNA that lie on the same chromosome pretty much right next to the gene. So here on this chromosome, we have the BRCA2 gene and the mutated BRCA2 gene right here in red. All right, and then the marker is right above it in blue. All right, so they're right next to each other. So remember, when you inherit a chromosome, you inherit the whole chromosome and all the genes on it. All right, generally we're only focused on one little gene on here, but this time we're going to be focused on two. So we're going to assume in this whole thing that if you inherit, and this is a big assumption, so sometimes that's why marker analysis isn't used, but it's a very high percent chance that you're going to, if you do inherit the marker, you're going to also inherit the BRCA2 gene. It's just that sometimes chromosomes change places, cross over, and stuff like that in meiosis. But these are so close to each other that most likely will have also the BRCA2 gene. So we're going to actually look for this marker. And if they have the marker, we're going to say that they also have the BRCA2 mutation. All right, and we're going to use gel electrophoresis to find out if they have this marker, because once we look at what the marker actually is, you'll see why gel electrophoresis is pretty easy to use to find that marker. All right, so the marker is just a non-coding section of DNA. There's tons of DNA in our cells that actually don't code for any proteins. It's just there. We're sure there does something, but we're not really sure what that parts of DNA does as of right now. All right, so what is a marker? All right, a marker is called a short tandem repeat or microsatellite. I'm always going to go with the short tandem repeat or STR because one, the STR tells you really what it is, a short tandem repeat, which once I tell you what it is, it's pretty obvious, and also STR is easier to say. So we have these short tandem repeats in man1, man2, man3. Well, short tandem repeats are repeats of certain sequence of bases that repeat so many times. So man1 here has CTA that repeats 5 times. Man2 has one that repeats 6 times, and man3 has one that repeats 7 times, and these can go on forever. And remember, gel electrophoresis measures DNA by length. The longer ones stay up towards the gel, top of the gel, shorter ones go to the bottom. So we can put the STR section of DNA into the gel, and then we can figure out how many repeats they have, and that's basically what we're going to be doing, or how long the fragment is. And then if you look here, this even tells you, so let's just assume this SNP is for the BRCA2 mutation. Man1, his 5 tandem repeats is going to be connected to the BRCA2 mutation here. This SNP, the single nucleotide polymorph, or the one base change there. So that's the BRCA2, and then these guys are normal. So what you would see on a gel is if we find that a person has 7 tandem repeats or a longer fragment, so if we look at our gel, and I'll explain this more in a second here with our actual example. So let's just say this is 7, 6, 5. So if we test another random man 4, and we find that he has 7 tandem repeats, then he is normal. If we find that he has 6 tandem repeats, he's probably normal. However, if we find that he has 5 tandem repeats, then he probably has the BRCA2 mutation, all right, because the BRCA2 is really close associated to that short tandem repeats. All right, and these are highly variable repeats, so they're also generally there's going to be very many different types of these STRs to test from, so they're not, we'll be able to find all the differences here. All right, so again, we're looking for these lengths of DNA on the gel, and then we're going to assume if we find one certain STR, we'll find the BRCA2 gene. So now I'm going to give you the overview, and then I'm going to show you how to do this specifically for Judy's case, and we're going to go a little bit in deeper than that. So if we look here, we have Bill, Laura, and Jennifer. We know we know these two have the BRCA2. Bill, remember, has passed away. So we know Laura and Jennifer have the BRCA2. So what we're going to have to do is using a standard curve and gel electrophoresis, we're going to have to figure out what STRs these two have in common, because if they have those STR in common, that means that's where that's the chromosome that the BRCA2 is. Because remember, Laura passed down has 2 chromosomes to give. She could even gave 1 chromosome with the BRCA2 mutation, or she could give 1 without. And Jennifer also inherits 1 from her mother and 1 from her father. So Jennifer's going to have either one of these and also another one from her father. So Jennifer's going to have 2 STRs that we're going to look for, and so is Laura. And we'll look at that in a second too. So the first thing we're going to have to do is determine which one of these STRs of the both of them do they have in common, and if they do have one even if they do have one in common, so to find out which STR the BRCA2, or the marker, which marker or STR the BRCA2 is attached to. Then we're going to do the same. We're going to also look at Judy and Diana's STRs. So then we're going to see if they have the STR that's associated with the BRCA2, then we're going to say that they also have the BRCA2 mutation. Alright, and we're going to do all of this using gel electrophoresis. So again, we're just identifying these STRs, and if these two have an STR in common with these two, then they most likely have the BRCA2 mutation. Alright, it'd be just like if we were looking at the BRCA2, the genes, and seeing if they are in common. Like if they all had the same sickle cell band, then they all have sickle cell. It's just a little bit different because we're just using STR. Alright, so here's their STRs. Now, a little bit different from this gel opposed to the test that we did before with our TACER gene, or if you would do it with sickle cell or Tay-Sachs or cystic fibrosis or whatever, is that we'll still use PCR to amplify it, and you still isolate the DNA and then do PCR, but the step that we skipped is restriction enzymes, because we don't need to cut it this time. The DNA is naturally differently longer based on the amount of repeats they have. Alright, so now what we're going to have to do here is actually normally we could just match the bands, and actually you could do that on this if you wanted to, and figure it out, okay, who has the BRCA2 gene. But we're going to go a little bit deeper, and we're going to actually find out the specific length. So we're actually going to find out how long these STRs are. So how long is this one? How long is this one? How long is this one? How long is it? And because we want to make sure for like a fact that Judy or Diana have the BRCA2 mutation, we don't want to misdiagnose them. So we're actually going to get some quantitative data here. So I'm going to show you how to get quantitative data from a gel. And we could do this with any type. We could do this for sickle cell, and in fact they do it for sickle cell and cystic fibrosis to make sure they get the right data. So what I'm going to do is I'm going to go through and show you how to read this and make a standard curve. So the first thing we're going to do is start with the DNA markers. And we're going to find out how far they moved from the gel, and we're going to assume, and we're going to make a curve that basically shows the trend of the correlation between how far they move and how big they are. And we're going to start with these DNA markers because we know how big these fragments are.